Thanks very much, Ali. And I do want to extend my personal thanks. Ali has done an incredible job coordinating this speaker series. And as she mentioned, our goal is to really bring together the scientific community in this virtual setting. A lot of people have been very hungry for the kind of discussions we have after conferences. And as we're transitioning from a pandemic to an endemic phase, I still think these lessons we've learned from virtual seminar series like this can really continue to strengthen the community. I'm very honored to be able to introduce Dr. Pat Megonigal. COMPASS, as mentioned, is a new project that DOE is supporting. Pat is our chief scientist, and he comes to us with a wealth of experience, but most significantly, I think it's important to note that he has been the Associate Director of Research for the Smithsonian Environmental Research Center, and he's the lead scientist for the research conducted at GROUP, the field experimental system that you see behind him on his backdrop. And he is also an incredible mentor and a peerless publisher. And with that, I will turn it over to Pat and let him spend the next 30 minutes telling us some stories about science. Thank you.
Well, thanks, Vanessa. Yeah, I really appreciate being invited to, share some of the work I've been doing with you, and particularly in this forum. I'm very excited about COMPASS as a project, really happy to be involved. And yeah, appreciate all of you who've joined the seminar today. So I want to start though by making sure you can see my slides. Are we good there?
It looks great, Pat.
Yeah, okay, good. I just always a good way to start a talk. So, all right. Well, yeah. So, as the title suggests, I'll be talking today about greenhouse gas emissions from coastal wetlands, but I'll have a particular perspective on this, which is the the way that plant microbe interactions tend to impart homeostasis on those emissions, and I'll explain what I mean by that in a bit. But I want to start with this kind of big picture. This is the present day imbalance in the global carbon cycle, so carbon dioxide. And what we see is in red, there's 10 petagrams emitted to the atmosphere, 90% of which are from fossil fuels. But the rate at which the atmospheric pool is growing is only half that amount, 5 petagrams per year, because the Earth's ecosystems are becoming larger sinks, 2.5 petagrams into oceans, which is the ocean acidification problem, and the other 2.5 into terrestrial systems. I'll point out a couple things. One is that the terrestrial number is the least well understood of these. And secondly, the terrestrial aquatic interface actually crosses both the terrestrial and marine side of this budget. Now, the main point I want to bring out on this slide is that the terrestrial sink is large. It's quite large. It's big enough that if you think about it a little bit, you might think, well, maybe if we were to manage terrestrial systems in different ways, adopt different management strategies, we could actually manipulate the rate at which CO2 is increasing in the atmosphere. And that, of course, is this concept of natural climate solutions. I chose this slide from the Nature Conservancy because they've been really out in front in trying to rigorously quantify what is possible in terms of natural climate solutions. And this is one such analysis for the United States published a few years ago. And what it's telling us is that if we were to adopt reasonable changes in the way we manage forests, agriculture, grasslands, and wetlands, we could offset about 1/5 of US CO2 equivalent emissions. Now, of course, forests are big here, but wetlands, even though they occupy a fairly small amount of the area, actually rate on this graph. They're a piece of the puzzle. And in particular, this includes coastal wetlands, which we often refer to as blue carbon ecosystems. In this case, they occupy less than 1% of ocean area, but store up to half of all ocean carbon. And so they are part of this conversation. Now, with that introduction to why we might care about this, I'm going to propose a thesis that I think actually crosses all terrestrial ecosystems and probably aquatic ones as well, which is that plant-microbe interactions place limits on the capacity of ecosystems to mitigate climate change. In other words, while natural climate solutions are important, there are some limits to how far we can go in that direction. And I underline limit because it's not that they can't mitigate. Again, there's just limits. And the reason for limits is that we have these opposing and linked biological feedbacks on climate that govern this. So on the left, we have plants that are taking carbon dioxide out of the atmosphere, providing a negative effect on radiative forcing. And on the other, we have microorganisms that are putting gases back into the atmosphere. And so depending on how any given perturbation, or in this case, global change factor affects one, we have to understand how it affects the other because they are in fact coupled. And the reason I put the word homeostasis in the title is because that's how I've come to think about this. Here's an example from some work that Melissa Pastor, Adam Langley, and I published a few years ago from the wetland behind me. And the x-axis is really carbon sequestration rate. And you can see that we're able to manipulate how much carbon is sequestered when we expose the system to elevated CO2 or to nitrogen or to the combination of those. But this is not a perfect relationship by any means, but there is a tendency when you do that to increase emissions of methane and nitrous oxide. And so this tendency tends to bring the manipulations back to this one-to-one line in terms of CO2 equivalence, which is what I'm calling homeostasis. So the work I'll present today that is meant to illustrate some of these interactions comes from the Global Change Research Wetland. It's on the western shore of the Chesapeake Bay. It has some particular characteristics important to the data that I'll present. It's in an organic soil, so over 80% organic matter, relatively little mineral matter deposited in this marsh. And it's dominated by a variety of species, but in particular, C4 grasses, including Spartina patens, and C3 sedges, Chenoplectus americanus. That's important because we do a lot of elevated CO2 research. That research started with this guy, Bert Drake, who, as he tells it, submitted 5 proposals to DOE before he got one funded. And this turned out to be one of the first 2 experiments in the field in situ involving elevated CO2. I sometimes illustrate the history of this with the famous Keeling plot, Mauna Loa curve. And you can see that this study, which is ongoing, started in 1987. It's now been running for over half the length of the Mauna Loa record. And the question that was asked then is whether elevated CO2 stimulation of plant production would be sustained in situ. I joined Bert actually in 1997, and then came to CERC in 2000. And in 2005, began adding related experiments at about 5-year intervals, culminating in COMPASS. And now you can see with COMPASS, the initial questions that we started off asking at this terrestrial aquatic interface have expanded considerably. In this case, we want to know about the mechanisms that control terrestrial aquatic interfaces across spatial and temporal scales and are important to improving predictive models. So I like to think that DOE's investment in the terrestrial aquatic interface that began in 1987 has been a bridge to this current and very ambitious project that we call COMPASS. Here's a little bit more about the experiments that are at the Global Change Research Wetland. Many of them have elevated CO2 as a common element, because as we learned certain things from the original experiment, we were able to pose new hypotheses that involved the influence of nitrogen, sea level, novel plant traits, which is how we think about Phragmites australis invasion, temperature. And we're now moving into the adjacent upland with Tempest, which is a storm surge and precipitation manipulation that will start in earnest in, well, this summer. Of course, there's a lot of people to thank for this work. These are all folks that have contributed to the data that I'll use today. I'll be drawing data from those experiments that were in red in the previous image. And of course, the funders here, especially DOE. So all of this work is related to this particular conceptual model that I'll take a moment to go over. The primary response variable that we focus on is elevation, and that's because it integrates the whole system. It integrates the plants, the microorganisms, the hydrology, and the geomorphology of the system. And of course, it's key to both carbon sequestration and the resilience of the ecosystem. Elevation can grow from the top up through sediment deposition. This is the above ground loop shown here, where above ground plant biomass stimulates sediment deposition. And there's a below ground loop, that's the balance between root productivity and microbial decomposition, which adds organic carbon, causing the soil to swell from the bottom up. Plants are key to both of these loops, and in fact, it's not just their productivity, but it's also the way they allocate that between shoots and roots that drive a lot of the dynamics in the system. And on the left is the list of global change perturbations that affect plant productivity, resource allocation to roots and shoots, and other processes that we're focused on. So now I'm going to go through 3 examples where these plant microbe interactions are important. And I'm going to present these for fun, if nothing else, in terms of dogmas, things that are widely believed but may not be true. The first of which is that elevated CO2 increases carbon sequestration. It's a reasonable thing to assume because it's pretty well known that elevated CO2 increases plant productivity. Here's an example of that from the original experiment that Bert started. And the area shaded in green represents the difference between the net ecosystem exchange of carbon dioxide at elevated CO2 in red and ambient CO2 in yellow over a 19-year period. So, it's reasonable to assume that at elevated CO2, we're getting increased plant production, and that's going into the soil, primarily increasing the soil pool, primarily the young soil pool. And so if that was the end of it, we would expect soil carbon to increase over time. In other words, the CO2 going in would be greater than that going out. But if that carbon input interacts with the microbial community to increase decomposition rates, we could actually see an increase in those rates and an entirely opposite pattern. We call this priming, and at an extreme case, you could actually lose organic carbon. We've seen that, but it could just also offset a lot of what you might have gained otherwise, and that is what I'm calling homeostasis. So this phenomenon of priming is well known and has been for decades, defined as an increase in the supply of organic carbon, causing an increase in the decay of relatively old or stable soil organic carbon. We've been working on this for quite a while, and usually starting in the greenhouse with that type of experiment. And at the Marsh, we have an advantage in that the C3 plants that respond so strongly to elevated CO2, that's the data I showed you in the 19-year data set a moment ago, have a very different isotope ratio than the C4 plants. And so if we take soil that is developed under C4 vegetation and plant C3 plants in it, what we end up with is a difference in isotope ratio between soil organic matter and plant organic matter that's 10 per mil. So when we capture CO2 coming off this system, both from plants and from soil, and take a sample of that, if its isotope ratio is say -22, we can calculate that 25% of that total came from soil organic matter. And we've done at least 3 of these experiments now, and it's very robust. And this is an example of what we've learned. You can see that elevated CO2 dramatically increases the rate of soil organic matter decomposition in this system. It causes priming. And what that means in the context of homeostasis is that more carbon in, because of increased plant productivity, also resulted in more microbial carbon out. More plant carbon in, more microbial carbon out. Now, this wasn't entirely a shock, and we interpreted it as carbon priming, but we also saw this relationship, which is that the plants grown at elevated CO2 also had the most root biomass, and the pots with the most root biomass had the highest soil redox potential, telling us there's oxygen, that they're enhancing oxygen movement into the soil, otherwise anaerobic soil. And from that, we coined this term oxygen priming, which has the same definition, but it's that your perturbation has increased the oxygen supply instead of the carbon supply alone. Now, as I said, we've done 3 published experiments on this at this point, but recently had an opportunity to do the most definitive test in the form of the salt marsh accretion response to temperature experiment. SmartX has a gradient design. That came a little early. SmartX has a gradient design, which is here at the bottom. You can see there's 4 levels of temperature ranging from ambient to 5 degrees C above ambient. And then we cross the extremes of that with elevated CO2. The experiment is a whole ecosystem warming experiment in that we warm the above ground vegetation with heat lamps and the soil to 1.5 meters with heating cables. We do this in 2 plant communities, 3 replicates in each. The heating started in 2016, elevated CO2 a year later. And all of this was done in the context, in a MODEX context, to improve ecosystem scale and Earth scale models. And it's really been led by Genevieve Noyce, who's a CERC research scientist. So credit for this data goes to her. Now, before I actually show you data from this, I have to establish a couple of things. First of all, there's an equivalency between elevation gain and carbon sequestration in this system. And I think this cartoon probably explained that pretty well. Secondly, elevation gain is very strongly coupled to root production in the system. Root productivity is the source of nearly all the organic matter, the mass that we're adding to the soil, causing it to swell. And we know this from some work that Adam Langley published back in 2009, and it's been a very robust observation for us. So with that as context, here is some data from SmartX. I want to start up top with the roots, and what you see in these open circles is the heating only treatments. And the first thing you'll notice is that our gradient design, which by the way we adopted so that we could detect nonlinear effects, is showing exactly that. We are seeing a very strong nonlinear response to temperature in which root productivity is highest at 1.7 degrees, basically the Paris Agreement. And once we get beyond that, it begins to fall. Elevation change is a nearly perfect reflection of that, which is what we came to expect from previous work. Now, the explanation for this nonlinear response is something I won't go into, but you can read about it in this paper that Genevieve Noyce published 2019. And then this elevation work is in press at Global Change Biology by Alex Smith, who's a student of Matt Kerwin's. So far, we have this surprising nonlinear result, but it's mostly following what we thought might happen. Now, when we cross this with elevated CO2, we get a big increase in root productivity, which again was expected from our previous work, although maybe a bigger increase than we normally see. The surprise came in the elevation data because there is no equivalent increase in elevation. And so from everything I've told you, and from everything we think we understand about this system, there's only really one good explanation for why there would not be a concomitant increase in elevation, and that is if priming had increased decomposition rates at the same time. So what evidence do we have of priming? And in particular, I'm going to talk here about oxygen priming. Well, we have this piece of evidence that comes from FTCIR data. This is data generated at EMSL, and we're very fortunate to have access to that facility to do this kind of work. What you see on the left side is a metric called the nominal oxidation state of carbon, which I won't explain here, but more negative values of NOSC, we call it, is telling us that the compounds have been subjected to more aerobic respiration. So in this case, these are NOSC values for the dissolved organic carbon pool in these treatments. And you can see that there's no some trends, but really no dramatic effects of temperature alone on the nominal oxidation state of carbon, but there's very large elevated CO2 effects. And that effect is in a direction that suggests increased aerobic respiration, and it's also related on the right to fine root biomass. Furthermore, when we plot this FTCIR data somewhat differently in this multivariate plot, the compounds at elevated CO2 are less aromatic, less lignin-like, and support this idea that they have decomposed in the presence of oxygen. So I take this evidence to be evidence of oxygen priming, supporting the idea that that's why we don't see a gain in elevation. We're seeing an offset of carbon sequestration, but it's all indirect. It's all indirect biogeochemical inference. And what we really need is measurements of redox potential. We have those now. And here's 2 years of it. There's a couple of things to notice. First of all is gut check. If we compare redox potential at 5 centimeters in dark green and black to 20 centimeters in light green and gray, the 5 centimeter redox potentials are higher. Okay, so now I can trust this data. Secondly, there's a lot of interannual variation here, particularly at the surface. Because this site is tidally flooded, that is not driven by depth to water table, or at least not primarily driven by that. So it's driven primarily by plant microbe interactions. But the point to take home for this talk is that at elevated CO2 in the dark green at the surface, we're seeing very large increases in redox potential, up to 200 millivolts. And so this, I think, is the smoking gun, that what we're seeing in SmartX is a case where elevated CO2 and temperature, although they increase plant productivity, that increased potential sequestration is offset by increased decomposition due to priming. Okay, now I'll move a little bit faster through the next two examples. Dogma number 2, flooding increases soil carbon sequestration. The more we flood a site, the less oxygen there is, the more carbon we store. Straightforward. We did a mesocosm experiment in the tidal creek at G-Crew. These are 75 centimeter tall pots filled with soil, some with plants, some without. And our simple hypothesis was that the wettest pots, the ones that are lowest in the water, would have the slowest rate of decomposition. But that's not what we found. So the wettest pots are the ones in blue and orange in this diagram. And you can see they had the highest rates of decomposition, while the driest pots had the lowest rates, opposite of what we expected. And we weren't really able to understand this until we, plotted the decomposition rates against above ground biomass. And so here again, we have an example where the flooding effects are subservient to the influences of plants on microorganisms. And because these plants grow better when they're wet, on the right side of this graph, those are the conditions under which soil organic matter decomposition is highest. So here again, more plant carbon in, more microbial carbon out, regardless of flooding. And dogma number 3, soil carbon sequestration mitigates climate change. Well, this seems reasonable, and actually is reasonable for the most part, I would say, in upland systems. But when we're talking about wetlands, tidal wetlands, we have this thing called methane. And what you see on the left are methane bubbles frozen into place in Lake Baikal, Russia. And in this instance, we understand that carbon sequestration is usually offset by methane emissions. And here's a couple examples of that. Couple examples where more plant carbon in means more methane out. This is a continental scale on the left for non-tidal wetlands. Higher net ecosystem production is strongly related to more methane emissions. And on the right, we draw from the SmartX experiment at a plot scale, gross primary productivity, and you can see this is a log scale for methane emissions related to methane emissions. And you can see that actually increases with warming. So these two are coupled, and this is the source of the homeostasis in terms of greenhouse gas balance in wetlands. And the mechanistic reason for it lies in this diagram. There's a relationship between the efficiency at which carbon is decomposed by microorganisms, one related to redox, and the amount of carbon that you can preserve in a system. So if we're over here on the left and we're preserving as much carbon as possible, we're also emitting a lot of methane. And if we do anything to try to lower the amount of methane emissions, we eventually find ourselves in this aerobic space where we're not preserving as much organic carbon. So the two are just fundamentally coupled in that way. Notice that there's a sweet spot in the middle where decomposition is dominated by sulfate reduction. And that is one reason that these marine blue carbon ecosystems are so attractive for carbon mitigation. Okay, so this unavoidable link only concerns the production of methane. And now I'm actually going to focus the rest of what I'm going to say about this particular piece of dogma on an exception, because I think the rule is established, and now I want to focus on the exception. And the exception is that we have these organisms called methanotrophs that also consume methane in the presence of oxygen. And so in this case, elevated CO2 increasing plant production and soil organic matter availability is expected to increase methane production and therefore methane emissions, causing emissions to go up. But we can offset that if plants are simultaneously introducing oxygen, increasing methane oxidation, in which case it might bring that curve back to homeostasis or maybe even bring it in the other direction. Bringing it in the other direction, I've always thought was theoretically possible, not really likely, but my view of that has changed. So we're back to the SmartX experiment. On the top is root-shoot ratio. I've already showed you some of this data. Temperature itself, which is all I'm showing now. If we look at the extremes, isn't having much of an effect on root productivity, but it greatly increases methane emissions because of warmer temperatures and more carbon into the system overall, probably through root exudates. But when we cross this with elevated CO2, we see a big increase in productivity. Root productivity, I've showed you this already, which we would expect to lead to more carbon and therefore more methane production, but instead it actually cuts methane emissions in about half. Something that you can only explain by increased methane oxidation, driven by plant-mediated oxygen flux into the soil. And so this sets up this very interesting way to think about wetland plants, because they're simultaneously adding carbon to the system, which is causing the soil to become more reduced, and they're adding oxygen to the system, which is driving it to become more oxidized. And depending on the plant, depending on the conditions the plant is growing under, one of these can dominate over the other. And as a result, we can get like very different responses under sometimes subtle conditions. Now, when we dig into this for the SmartX experiment, we discovered this phenomena, which is on the left, Spartina patens. As Spartina patens biomass increases, methane emissions increase with it. This is a log scale. That's how I've always thought most plants work, most wetland plants. But then we've got the sedge on the right, where as biomass increases, methane emissions actually go down. So it seems like there are certain plant species or certain plant traits that we can use to classify plants like wetland plants into one of these categories or another. And we know that these plants in wetlands are ecosystem engineers. And if that's the case, then plant traits are their tools. And there's a lot of plant traits that we typically recognize in terrestrial systems. These plant traits are leveraged heavily for models of all sorts, including Earth system models. And Genevieve and Peter and I and others are advocating that we need plant traits like these in red at the bottom that relate specifically to the influence of plants on redox potential. Can we classify these wetland plants as either net oxidizers or net reducers? And if we can, it should help us reach our COMPASS vision, this robust mechanistic understanding of terrestrial aquatic interfaces to support process-rich modeling. In other words, this may help us get from the condition above in the top illustration showing the current state of Earth system models in which land is connected to oceans with a single pipe that we call a river, to the bottom illustration where those rivers flood into wetlands, drain back, have all sorts of benthic interactions, shallow groundwater. In other words, much more realistic depictions of these processes. So to summarize, plant-microbe feedbacks tend to establish a homeostasis in terms of greenhouse gas balance in terrestrial ecosystems, and probably to some extent in aquatic ones as well. These interactions are particularly important in wetlands because here we have systems that produce methane, and that very much works into the greenhouse gas balance. Despite these interactions, I don't want to leave this seminar without saying that unmanaged terrestrial ecosystems are indeed mitigating climate change through increased carbon sequestration. But my point is that there's just some limits to that we need to understand. And if we can capture these plant, microbe, soil, I would add water interactions and predictive models that can inform us about both the potential and the limits for terrestrial ecosystems to mitigate climate change, then we will have succeeded. So thank you very much. And with that, I'm finished.